Today we'll do Biomolecules class 12 chapter, and in which number of important things related to the bio. It is easy for the bio students and little bit tough for the non-bio student, that is non-medical students. So first of all, we'll do carbohydrates. So carbohydrates are the primarily produced by the plants and form large group of naturally occurring compounds and were considered as hydrates of the carbon. So this was the old definition which was given, like for example, if you have C6H12O6, like C6H12O6, if you simplify it, it is CH2O, where this is made up of carbon, hydrogen and oxygen, and hydrogen and oxygen, they are in the ratio of water. But this definition was failed when the rhamnose was discovered, and in the rhamnose the formula was C6H12O5, where oxygen and the hydrogen, they were not in the ratio of H2O. So this definition was failed. Then the new definition came, that is carbohydrate may be defined as optically active polyhydroxyaldehyde or ketone or the compound which produces such units on hydrolysis. Now what is the meaning of optically active? Means that chiral, means that compound should be attached with the 4 different atom or group of atom, and aldehyde and ketone will be the compound and which produces such units on hydrolysis. Like again the example of glucose if I take, and in the glucose which is C6H12O6, if we'll write, then carbon, 6 carbons are there, 1, 2, 3, 4, 5 and 6. On the first carbon there is aldehyde, and on the third carbon OH is on this side. So this is H, OH, this is H, this is H, OH, then H, OH, H, H, and this is OH. So this is polyhydroxy because number of OH groups are there. Aldehyde, that is CHO, and it, like suppose if we'll take the example of fructose, there will be the ketone. So polyhydroxyaldehyde, example is glucose, polyhydroxyketone, example is fructose, and it gives such compounds, such units on hydrolysis. Now carbohydrates, they may be sugary or non-sugars. The differences are, like in case of sugar, that is sweet in taste, and non-sugar, they are tasteless. Sugar soluble in water, non-sugar insoluble in water. Sugar crystalline, that is regular arrangement, and non-sugar, they have irregular arrangement, so they are amorphous solids. Now we have classified carbohydrates into 3 groups. First is monosaccharide. Monosaccharide which you cannot hydrolyze it further to give simpler unit of polyhydroxyaldehyde and ketone. Example glucose, fructose, they are simplified. You cannot further convert it into any other carbohydrate or simple monosaccharide unit. Mono means single, saccharide means sugar. Then is the disaccharide. Disaccharide which yield 2 or 2 to 10 monosaccharide unit. Like if the 2 saccharide unit, disaccharide, 3 tri, 4 tetra and so on. Like example for, mainly I have given the disaccharide example which is there in the syllabus also, that is sucrose, lactose and maltose. Then is the polysaccharide which yield large number of monosaccharide unit, for example starch and glycogen. So if we have divided into 3 now, monosaccharide, disaccharide and polysaccharide. Now monosaccharide, we are classified into on the basis of number of the carbon atom and the functional group between them. Like if the monosaccharide contain aldehyde group, as I told you, is a glucose, where the CHO is there, we call it as aldoses, and when the ketone group is there, we call it as ketoses. Now first of all, we'll do the glucose which is a monosaccharide, and the method of preparation from the sucrose is, like you have C12H22O11 and we'll put water into that, it give H positive, and this is C6H12O6 plus C6H12O6. So this is your sucrose which we call it as cane sugar also, and when you hydrolyze it, it changes into glucose and it changes into fructose. Another method of preparation is from starch. Now in this case the C6H10O5 whole N, that is the starch unit, and we'll put N water in the presence of H positive, 393 Kelvin temperature and 2 to 3 atmosphere pressure. Then this starch unit you will hydrolyze it, and this will give N mole of glucose, that is C6H12O6. Next, what is the structure of the glucose? Structure of glucose, though I have already explained you, again I am explaining, that is C6H12O6. In this again 1, 2, 3, 4, 5 and 6 carbon, and on the third OH is on this side. Here is CHO, here is H, OH, then H, OH, then H, OH, then H, H and OH. So this is for the structure of the glucose. Then what are the reactions of the glucose? First of all, the reaction with HI, like if we'll take glucose, if I'll write like this CHO and or rest of the 4 carbon, I'll write like CHOH whole 4 and CH2OH, and if I'll add HI and I'll heat, it forms N-hexane, that is CH2 whole 4 and then CH3. So this will form N-hexane, though iodohexane also form, but this is the main product, so we are writing this only. This is N-hexane. Then is a reaction with the NH2OH, that is hydroxylamine. If we'll take CHOH whole 4 and then CH2OH, and if I'll add hydroxylamine, that is NH2OH, then water moves out and this forms CH double bond NOH, and this is CHOH whole 4 then CH2OH. This is known as glucosazyme, or you can say glucose oxime, you can say. So glucose oxime will form. Similarly, when we react with HCN, that is CHO, CHOH whole 4, CH2OH, and if I'll add HCN, then it gives CHOH and CN. This forms CHOH whole 4 and CH2OH, and this forms glucose cyanohydrin. Then is reaction with the bromine water. If we'll take glucose again, that is CHO, CHOH whole 4, CH2OH, and if we'll react with the bromine water, if I'll add, then one of the aldehyde, because this is an oxidizing agent, will change into carboxylic acid, that is COOH, CHOH whole 4 and CH2OH, which we call it as a gluconic acid. Then reaction with the acetic anhydride. If I'll take, now I'll write it in the open form because it would be easy for 1, 2, 3, 4, 5 and 6. Now third OH is on this side, H, OH, H, OH, H, OH and CH2OH. I will react with acetic anhydride, that is 5 mole of acetic anhydride if I'll take. Now there are 5 H, 1, 2, 3, 4, 5. So it will cleave like this and it forms 5 mole of CH3COOH plus, this is your CHO, CH, OCOCH3, CH, OCOCH3, CH, OCOCH3, CH, OCOCH3, then CH2, OCOCH3. Now the product which is formed here is known as glucose pentaacetate. This is glucose pentaacetate. Next is the reaction with HNO3. That is, again we'll take glucose CHO, CHOH whole 4, CH2OH. It will react with HNO3, which is an oxidizing agent. Now the first will change into carboxylic acid, and the last one is again will change into carboxylic acid. This we call it as glucaric acid. Next is the D-glucose and the L-glucose. Now D and L-glucose is that earlier we gave the term on the basis of the structure of glyceraldehyde. Now in the glyceraldehyde, like you have CHO, H, OH and CH2OH. If OH is on the right side, we call it as D, and if OH is on the left side, then we call it as L-glucose. So this way we can say D and L-glucose, right? But what about the glucose? Glucose we have 6 carbon, 1, 2, 3, 4, 5 and 6. On third OH is here, H, OH, then is H, OH, and this is your CH2OH. So we'll say on the basis of 5th carbon, if in the glucose carbon 5, OH is on the right side, then we call it as D-glucose. If OH is on the left side, we call it as L-glucose. In the nature mainly it exists as a D-glucose. Now is the cyclic structure of glucose. Because there were some limitations in the open chain structure of the glucose, then we needed the cyclic structure of glucose. So despite having aldehyde group, glucose does not give 2,4-DNP test. Now we just saw that the glucose possesses CHO group. So these tests glucose should give. It should give 2,4-DNP, Skiff reagent test, but it does not form sodium hydrogen sulfide addition reaction with NaHCO3. So they are not giving some of these reactions which glucose should give because it possesses aldehyde. Similarly, pentaacetate of the glucose not react with the hydrogen sulfide, again indicate the absence of free CHO group which open chain structure could not explain. Another limitation was that glucose is found to exist in 2 crystalline form, alpha and beta. Alpha form is obtained by the crystallization from the concentrated solution of glucose at 303 Kelvin, and beta is obtained from the hot and the saturated aqueous solution at 371 Kelvin. So there are 2 different glucose forms, but in the open chain structure there is only one form, which again open chain structure could not explain. Now to explain it, there was one more new terminology, you can say anomers. Now how to draw the anomers? Like you have, again we'll draw the glucose, but this is the open form like this, 1, 2, 3, 4, 5 and 6. On the third side, OH is on this side. Now all the H on one side and OH on other side. Now what will happen, that you have 1, 2, 3, 4, 5 and 6. On the 5th carbon, this will shift this side and these electron will shift to this oxygen. Now this reaction will be at equilibrium. This is your CH. Now this becomes OH. This is H, OH. Third OH this side, H, C, H, OH, CH, and through oxygen they link and they form CH, H, OH. Similarly, we'll draw the equilibrium structure this side, but the only change will be OH will be on this side instead of that side. Here OH is on this side, here OH is on this side. So it is CH OH, COH, H, CH, OH, CH, and through oxygen they will link. This is known as hemiacetal form of the glucose. Now in this case, if OH is on this side, this is known as alpha D positive glucose. Alpha means that OH is on the right side. This is your beta D positive and then glucose. So this is alpha glucose and beta glucose. Next is the pyranose structure. Pyranose structure is, you can say that is Haworth projection formula. This we also call it as Haworth projection formula. That was hemiacetal form, and this is a fully cyclic form. So this is the Haworth projection formula. Now what is the rule of the Haworth projection formula? Again, now what we are doing, we are making for the alpha D glucose. This is H, OH. This is OH, H, H, OH, then H, and through oxygen they are linking, and this is CH2OH. Pyranose form means that we are making the 6-membered cyclic ring structure with the oxygen. Now in this is the pyran form, in which we'll put the glucose formula in this. Now in this, whatever is written here, that will be on the lower side, and whatever is written here, that will be on the upper side. Oxygen will always be at the upper right hand carbon. This is this oxygen. Now we'll start with 6. This is the 6 carbon will always be here. So this is your 6, 5, 4, 3, 2, 1. So the 6 carbon is here. This is H. Then this is the 4th carbon, and that means this one. H will be on the upper side, OH will be in the other. Then OH, H, H, OH, then is H and OH. This was the alpha glucose. This is alpha glucose. In alpha glucose, always the OH will be on the lower side. So this is alpha. And if you draw the beta, then the OH will be on the upper side, on the carbon 1. And this, when the OH is on the upper side, this is beta D glucose. Next is the fructose enomer form. Now the fructose, first of all, we will draw the structure of fructose. Carbon, the formula for the fructose is again C6H12O6. 1, 2, 3, 4, 5 and 6. Now in this case, again third OH this side, because ketone is there, it is on the second side. Now we'll write all the H on one side. H, H, H and H, and OH on other side. OH, OH, OH and H. Right? Now what we'll do, there is also OH. We'll make the anomers of the same. Again C5 and C2. This will move here and this will come here. Now we'll draw the anomer of the same. Now this is your 1, 2, 3, 4, 5 and 6. In carbon 2, this is OH and this is CH2OH. This is C, OH, H. This is H, this is OH, then H, and through oxygen they will link, and here it is CH2OH. Now we'll see that carbon 2, if the OH is on the right side, then we call it as, alpha D fructose. Now similarly we'll do the anomers of this. just will change the position of the OH. Now OH is on this side, CH2OH this side. So this is your 1, 2, 3. Third OH will also remain on this side, H. Fourth HOH. Fifth H. And through oxygen again they link, and this is your CH2OH. So this is your beta D fructose. Beta D fructose. So anomers are these two are the anomer which have the same molecular formula but the different arrangement only at the carbon 1, that is called anomers. Next is the furanose form of the fructose, like we did the pyranose form in case of glucose. Now we'll draw the fructose furanose form. So first of all we'll draw the hemiacetal form, that is your C. Suppose CH2OH, OH, that is alpha we are making, so OH is on this side. This is 1, this is 2. Then third OH is on this side, H. Fourth H, OH. Fifth H, and through oxygen they link, and then CH2OH. Now in this we'll draw the furanose form. Now here this is your 6, 5, 4, 3. Now the sixth will always be on this side. Now whatever is here will be on the upper side. So here it will be CH2OH and OH. Then third OH, H, H, OH. So on the lower side if OH is there, you remember that is alpha, and if the same way I'll write OH on the upper side and CH2OH on the lower side with the same structure, then this is beta form. Next is the disaccharide. With a 2 monosaccharide unit, they join with each other together by an oxide linkage formed by the loss of water molecule. Such a linkage between the 2 monosaccharide unit through oxygen atom is called glycosidic linkage. So the definition says that 2 monosaccharide unit, they join with each other through oxygen atom and is called glycosidic linkage. So mainly we have 3 examples of the disaccharide, that is sucrose, which always after the hydrolysis gives glucose and fructose, maltose gives glucose and glucose, and then you have your lactose, which on hydrolysis gives glucose and galactose. Now first we'll do the sucrose. First of all, the method of preparation of the sucrose. In this method of preparation, as well as you can say that the reaction of the glucose, they are mainly made from the C1 of alpha glucose and the C2 of beta fructose. So when the 2 monosaccharide unit, they are held together by the glycosidic linkage between the carbon 1 of alpha glucose and carbon 2 of beta fructose, then the reducing group of the glucose and fructose are involved in the glycosidic formation, and this is called non-reducing sugar. This is the only, like in the exam also, if they'll ask you name one disaccharide which is non-reducing, then you can give the example of the sucrose. Now we'll draw the structure how the C1 of alpha glucose and C2 of beta fructose is forming. Now Haworth projection formula the same way. Now the alpha glucose, first make this structure, then CH2OH on the upper side, third OH on the upper side, and alpha if it is there, then the OH will be on the lower side. So this way, and then draw rest of the hydrogen and oxygen. Here is H, here already OH on the upper side, H, OH, H, H, here OH on the upper side, H and OH. So this is your alpha glucose. Now we'll draw the beta fructose. Now beta fructose, and we'll write in the reverse way because we need C2 of beta fructose to link with this OH. So how we'll write? We'll write like this. In this CH2OH, which will link here. OH is there, so the water will move out, and this will be your glycosidic linkage. Glycosidic linkage. Here is your H, OH, OH, H, H, and CH2OH. So this is the fructose, and this is glucose, where the linkage will occur and removal of water will occur, and the linkage is known as glycosidic linkage. Now, what is the inversion of the cane sugar? Inversion of the cane sugar is when the sucrose gives glucose and fructose. Initially, the angle of rotation is 66.5 degree, that is in the plus, this is dextrorotatory. But when you do the hydrolysis, angle changes into plus 52.5 and minus 92.4. Because minus angle, alpha angle of rotation is more than the dextrorotatory, the whole solution will change from dextro to levorotatory. And this product named is known as invert sugar, or the process is known as inversion of the cane sugar. Next is the maltose. Maltose is made up of two glucose unit and both are alpha. Carbon 1 of one glucose unit linked with the carbon 4 of another, and this is reducing sugar. Earlier sucrose I have already discussed, that was non-reducing, this is reducing sugar. Now in this case we'll draw the 2 alpha D glucose unit. Again we'll follow the same procedure, 6 CH2OH on the upper side, OH on the upper side, and because it is alpha, OH, H, H, OH, H, OH. OH, H. And the water removal will occur, H2 and O2, and through oxygen they will link with each other. And this kind of the linkage which is linked through the oxygen, that is again we call it as glycosidic linkage. This is glycosidic linkage. Next is the lactose. It is commonly known as milk sugar and is composed of beta D galactose and beta D glucose. Carbon 1 of the galactose and carbon 4 of the glucose, they will combine, and it is also again a reducing sugar. Now we'll draw the structure of galactose and glucose. There is only the minor difference between the galactose and glucose. CH2OH, H. Beta every time OH on the upper side, remember. Third OH on the upper side. But here we are making the structure of galactose, so third and fourth both OH will be on the upper side. This is the key point to remember in the case of galactose. This is galactose. Now we'll take carbon 1 of galactose and remember carbon 4 of glucose. Now we'll draw the glucose structure here, CH2OH. Then your OH on the upper side, and because this is also beta, so OH on the upper side, then H, OH, then is H, OH, and the removal of water will occur like this, H and OH, and they link like this, zig link. And this is because this is beta galactose and this is beta glucose, so the linkage which is there is a beta glycosidic linkage. Beta glycosidic linkage. This is the beta glycosidic linkage. Next is a polysaccharide. Polysaccharide contain large number of the monosaccharide units joined together by glycosidic linkage. And we have divided into two, amylose and amylopectin. Amylose is water soluble, amylopectin is insoluble in water. Amylose contains 15 to 20% of the starch. It contains 80 to 85% of the starch. Amylose is long unbranched chain of 200 to 1000 alpha D glucose and C1-C4 linkage. While in case of amylopectin, alpha D glucose unit, C1-C4 and C1-C6, both straight chain as well as the branch linkage is there. Now first we'll draw the structure of amylose. The structure of amylose is similar to that of maltose, but the number of the, as we have seen, that the glucose unit would be more. Now in this case, H, O, H, H, and this side will show it like this, which indicate that yes, the chain is continuing on both the side. Now we'll draw the rest of the unit. CH2OH, H, OH, H, OH. OH, H, OH. This is OH, H, H and OH. So such kind of the long chain, if they will continue and they are having C1, C4, this is 1, this is 4, this is 1, this is 4, C1, C4 linkage, that is called amylose. Next is the amylopectin. In the amylopectin, there are two different kind of the linkage. One is the same as we did in case of amylose, C1, C4 linkage, and another one is a C1, C6 linkage. Now this is C1, C4. Now this is H and O and CH2, H, this way, and now again H, O, H. H, O this side, H, O this side, and H, O this side. So here it is CH2OH, H, CH2OH, H, OH, H. That again the same way will complete. HOH, OH, H, OH. CH2OH, H, OH, H, H and OH. So this is your C1, C4 linkage. This is C1 and C6 linkage. So two different kind of linkage, C1, C4 and C1 and C6. So these two linkages are there. Next is the cellulose, which you know that occurs chiefly exclusively in the plants, and the predominant constituent of the cell wall of the plant cell. It is a straight chain polysaccharide composed of beta D-glucose unit, in which C1 of one glucose combined with the C4 of the next glucose unit. But here the important point is that both are beta D-glucose unit. Now to draw the structure, will first of all one beta D-glucose unit will make CH2OH, H. Now OH, H, H, OH. Now we'll draw it like this, that the chain is continuing. This is H and OH. This is carbon 4. Now we'll draw the second unit in such a way that the carbon 1 is coming exactly, and because it is beta OH on the upper side, then CH2OH, H, H, and we'll write like this, the chain is continuing on this side, OH, H, H and OH. Now H from here and OH from here, water moves out. Now in this case, through oxygen they'll link, and this is 1, this is 4, and the linkage is again beta glycosidic linkage, beta glycosidic linkage, beta glycosidic linkage. Now glycogen. Glycogen, the carbohydrate stored in the animal body as glycogen, and we also call it as animal starch. Structure similar to that of amylopectin, rather highly branched. So if in the exam the structure will come, you can draw the structure of amylopectin, and it is highly branched. It is also present in the liver, muscle, brain, and during the fasting or you are doing lot of exercise, then the body need glucose, and enzyme breaks this glycogen down to glucose. This is also found in the yeast and fungi. Now, what is the importance of the carbohydrate, or you can say the uses of the carbohydrate? They are essential for the life in the plants and animals. They provide energy for the functioning of the living system. They act as biofuel. They are reserve food material in the plant. It is a chief structural material of the cell wall of bacteria and plant. It is an essential component of RNA and DNA. So, in case if they'll ask the uses, so these are the few uses of the carbohydrate which you can mention. Next is the protein. Protein basic unit is alpha amino acid. This is the alpha amino acid, which is the basic unit of the protein. They are most abundant biomolecule of the living strain. Like in our body also, like hair, skin, everything is made up of protein. So that is a very important constituent in our diet also. And the chief sources are milk, cheese, pulses, peanuts, fish, meat. and they are also required for the growth and the maintenance of the body. Protein, the meaning of the protein, which has come from the Greek word proteos, means that prime importance, that in our body the protein is the prime importance. So you can see in the diagram also, basic unit is amino acid, which is forming peptide. Peptide is forming protein. So basic unit is alpha amino acids. Now amino acids both contain amino group and carboxyl group, that is NH2 group and COH group. Alpha amino acid are obtained on the hydrolysis of the protein. All the amino acid, they have trivial names. Trivial names with the 3-letter word symbol. And the, like for example, if we'll say glycine, then the 3-letter is GLY. Similarly, it can be 1 letter, it can be V also or G also. And the structure is like you have amino on one side and carboxyl on another side, and H and R. So this is amino and this is carboxyl group. So they possesses both acidic as well as basic groups. Other properties or the characteristics of amino acid, they are colorless, crystalline solid. They are water soluble because they are behaving like a salt. Like you have amino also and carboxyl also, they are behaving like a salt. They are water soluble, and like salt they have high melting point also. They behave like simple salt. This behavior is due to the presence of both acidic and the basic group. And in aqueous solution, they accept the proton and give the dipolar ion known as Zwitter ion. This is the neutral but contain both positive and negative charge. Like you have suppose R, CHNH2, C double bond O OH. Now in this case, H will move like H positive and will come here on the lone pair of the nitrogen. Now in this case, they'll form R, C, H, this becomes NH3 positive and this becomes a COO negative. So this is a dipolar ion which behave like a Zwitter ion. This is a dipolar ion which behave like a Zwitter ion, where they behave like a salt also, positive ammonia and ammonium ion, and the CO negative, that is the acidic carboxylic ion. So they are dipolar ion, they possesses both the properties. Next is the D and L amino acids. Like there was D and L glucose, this is D and L amino acid also. So depending upon the position of NH2 group, like you have NH2 here, COH here, H here, R this side. If your NH2 is on the right side, then we call it as D. And if NH2 is on the left side, then we call it as L. So if it is on the right side, D, on the left side, this is L. Except glycine, all amino acids are optically active. This you need to remember, because in the exam also the question can ask, because there are like 20 amino acid mainly which is found in our body, and glycine is only that amino acid which is optically inactive. Sorry, this is except all. So glycine is optically inactive. So because there is H, and here it is NH2 and here it is COOH. So except glycine, all the amino acid, they are optically active. Only this is the structure of the glycine which is optically inactive. Next, what is the structure of the protein? Structure of the protein, when the two amino acid unit, they join with each other, like you have COH and you have NH2 here. Then again you have NH2 here, suppose HR, and COOH this side. Now COH, OH move from this side, H moves from this side. Now the water removal will occur, and in this case NH2, CH2, CO, NH, C, H, R, COOH. Like this is your glycine and this is your valine. And this is the peptide bond which is formed between the two amino acid unit. So this is the peptide bond. So when two amino acid unit, they join with each other with the removal of some simple molecule like water, that kind of the bond which is called peptide bond. Now the classification of the protein. They are mainly classified into two types, fibrous and globular. Fibrous, when the polypeptide chain run parallel and held together by hydrogen bond, disulfide bond, then the fiber-like structure which is formed is called fibrous protein, straight chain. But when you coil it, like the hair, if you'll coil it, it gives a coil-like structure, spiral coil-like structure, that is called the spherical shape which is there is called globular protein. Now, if the straight chain is there and if the coiled chain is there, the straight chain will be insoluble in water because the bonding will be strong and they will be stable toward the change in the pH and temperature. While in case of globular protein, they are soluble in water and they will be unstable toward the change in the pH and temperature. Fibrous protein, example keratin, myosin, and globular protein, example insulin, albumin, etc. Now, this is the structure and the shape of protein which can be classified at 4 different levels. Like you can see, this is the first one. This is the primary structure, which is the basic structure of the amino acid. And when it further coils up, then it gets converted into the secondary structure. And further coiling gives tertiary structure. And further coiling, if it will do, give the quaternary structure. So, in our body, like this is the example for hemoglobin. And in our body, that protein is existing in the quaternary form. This is the actual form in which the protein is existing in our body. Now, this is the primary structure of protein. Primary structure means like you have amino acid link in the straight chain. Like proteins may have one or more polypeptide chain, and these amino acid linked with each other in a specific sequence. The important thing is a specific sequence, because if the sequence will change, they'll lead to change into entirely new different protein. It creates the entirely different protein. Like for example, there is a disease called sickle cell anemia, where the shape of the RBC changes. It changes into the sickle cell. So that is your primary structure of the protein. Then is the secondary structure of the protein. It refers to the shape in which long polypeptide chain exists. And it is of two types, alpha helix and beta pleated. This is the alpha helix sheet and this is your beta plated sheet structure. And both are the types of alpha secondary structure. Now, in the alpha helix, you can see that, and both these structure arises due to the regular folding of the backbone of polypeptide chain. Why they folded? Because the hydrogen bonding occurs between C double bond O, that is carbonyl, and NH group of the peptide chain. So, if they'll form the hydrogen bond, then the primary structure, which was the straight structure, will coil it up, and they can give the alpha helix and beta plated. Now, secondary structure of protein divided into two, alpha helix and beta plated. A polypeptide chain form the all possible hydrogen bond by twisting into right-handed screw-like structure, this way. So, this is alpha helix structure in which suppose NH is from this side and the carbonyl from the next side, and they form hydrogen bond with each other. Similarly, you have NH this side and the carbonyl that side, they form hydrogen bond with each other. So, the first CO and the fourth NH, they form hydrogen bond with each other, and they changes from the straight chain to the coil like structure. There is a beta pleated sheet structure. Like all the polypeptide chain, you stretch and you lay side by side like this. When you lay side by side like this is the pleated fold of the drapery, where all the NH on the one side and all the carbonyl on the other side. And this NH from the one side and the carbonyl from the other side, they form hydrogen bond. So, this way they are connected. So, I am reading it. All the polypeptide chains stretched to the maximum extension, laid side by side, held together by intermolecular hydrogen bond, and these resemble the pleated folds of drapery. That is called beta pleated sheet. This is the tertiary structure of the protein, which represent further overall folding. Like primary was straight, then you coiled up, secondary. Then again you coiled up, that is give the overall folding of the polypeptide chain, and it give rise to the two major shapes, fibrous and globular. And the forces which are responsible for the secondary and the tertiary structure of protein are hydrogen bond, disulfide linkage, van der Waals, and electrostatic force of attraction. So, these four forces are mainly responsible for the formation of the tertiary structure of proteins. Then quaternary structure. and in this form it is exactly present in our body. They are composed of two or more polypeptide chains. They are referred to as subunit. Like in the hemoglobin, we have two alpha chains and the two alpha beta chains. Like your alpha chain, they are made up of 141 amino acid, and beta they are made up of 143. And the arrangement of these units with respect to each other is known as quaternary structure of proteins. So you coil up and you get the structure. Now, what is the denaturation of protein? D means change, naturation means nature of protein. Each protein, they have their, in the native state, they have their subjected to the chemical change, they have their particular pH temperature. But if we do some changes in the temperature or the chemical change like pH, hydrogen bonds are disturbed and they get uncoiled, unfold, and they loses their biological activity. During the denaturation, secondary and the tertiary structure destroyed, but the primary structures remain intact. So change in the nature, like curdling of milk, right? So these are the example. So they are in the quaternary form. They uncoiled, secondary coil, uncoiled, tertiary uncoiled, and the primary remains intact. So this is known as denaturation of the protein. Next is the vitamins. They are the organic compound required in diet in small amount to perform specific biological function and for normal maintenance of optimum growth and the health of the organism. We all know that vitamins play a vital role in our body. And now they are divided into two types, water soluble and fat soluble. Water soluble, they are soluble in the water, and all the vitamins like vitamin B and C comes in this category. Fat soluble, which are soluble in the fat, not in the water, like example A, D, E, K. These are the mainly fat soluble vitamins. Now these are few vitamins and their sources in the deficiency disease. Like vitamin A, their main source are fish liver oil, carrot, butter, milk. But if you will not take it in the sufficient amount, they can lead to the disease called xerophthalmia, that is hardening of cornea of the eye, and night blindness. You will not be able to see in the night. Next vitamin is B1 and the sources is yeast, milk, green vegetable. The deficiency disease beriberi, that is loss of appetite, you will not feel hungry, and obviously if you will not take proper diet, it leads to the retarded growth. Vitamin B2, that is sources are milk, egg white. It leads to the chillosis, that is fissuring of the corners of the mouth and lips. That happens normally in the winters. Digestive disorder and the burning sensation of the skin. Vitamin B6, that is yeast, milk, egg yolk and cereals. Convulsions means you feel like headache or convulsions like you'll not feel healthy, something related to the brain. Next is vitamin B12. Sources are meat, fish, egg, curd. And the deficiency is pernicious anemia, where the RBC deficiency is there in the hemoglobin. Vitamin C, if you will not take vitamin C, then it leads to the disease called scurvy, means bleeding gums, and the sources are citrus fruit like amla, green leafy vegetable. Vitamin D, the main source is exposure to sunlight, fish, egg yolk, and if you will not take it properly, the diseases can be rickets, that is bone deformities in children, osteomalacia, that is soft bones and joint pain in adults. Vitamin E, the sources are vegetable oil like wheat oil, germ oil, and the disease can be increased fragility of RBC and muscular weakness. Then is vitamin K, green leafy vegetable, and the deficiency disease will be increase the blood clotting time. Next is the nucleic acid. The particles in the nucleus of the cell responsible for the heredity are called chromosomes, and which are chromosomes are made up of protein, and another type of the biomolecule is called nucleic acid. So nucleic acid is basically responsible for transferring the character from the parents to their offsprings, and they are mainly of two types, deoxyribonucleic acid and the ribonucleic acid. Now nucleotide. Nucleotide is formed by the combination of three components. Base, that can be your ribose sugar, these are basically the nitrogenous base, and then the pentose sugar. This pentose sugar can be ribose in case of RNA, and it can be deoxyribose, that is 2-deoxyribose in case of DNA. 2-deoxyribose in case of DNA, and the phosphoric acid, that is your H3PO4. When you'll combine all these three, they lead to the formation of nucleotide. Now nitrogenous base. Now we'll draw the structure of the first nucleotide. In this we'll draw the structure. In this we have H, OH, H, OH, then H, and then CH2OH. Then here is H, and here you can write nitrogenous base. Nitrogenous base are mainly 4 type, that is adenine, represented by A, then guanine, that is with G, then thymine, that is represented by T, and cytosine, that is represented by C. And adenine always form bond with the thymine, doubly bonded, and guanine form bond with the cytosine. In this there is phosphate unit, that is P double bond O, OH, OH, OH. Either you write OH or O negative. Then the water is not here and they'll combine through the oxygen. So this is nitrogenous base, this is sugar, and this is your phosphate unit. So all these three when combine with each other and they form nucleotide. There is a nucleoside, and the nucleoside, the difference between the nucleotide and the nucleoside is, in the nucleoside there are all three things, sugar, base, phosphate, while in case of nucleoside there is sugar and only base is there, there is no phosphate unit. So here is H, here is nitrogen base, then is H, OH, H, OH, then H and CH2OH. So only nitrogenous base is there, and this is your sugar unit. Then how we form the dinucleotide? Then two dinucleotide, they combine with each other in the presence of phosphodiester linkage. So here is your sugar first. This is sugar, where we'll put your H, H, OH. Here is H. There is a nitrogenous base. This is base. Then H, CH2, O, P, double bond O, O negative. This is one nucleotide unit. Now the second nucleotide unit will join like this, O, P double bond O, O negative, O, CH2, here is nitrogenous base. So this is 1, 2, 3, carbon 3. This is 1, 2, 3, 4, 5. So carbon 3 and carbon 5, they link with each other, and this linkage is known as phosphodiester linkage. This is phosphodiester linkage. We have studied now three different kind of linkage. One was glycosidic linkage between the two monosaccharide unit. Then is a peptide linkage between the two amino acid, and this is your two nucleotide as there. This is one nucleotide, and the another nucleotide, the linkage is known as phosphodiester. This is another nucleotide, and the linkage is known as phosphodiester linkage. Then the question can be asked that explain the term that the two strands in the DNA are not identical but they are complementary. So in the helical structure of DNA, hydrogen bonds are formed. Adenine always form bond with thymine, and cytosine, they always form bond with the guanine. Like in the double helical structure, if you draw in the double helical structure of DNA, which was given by Watson and Crick, in this, if you draw this double helical structure, of the DNA, where this is 5' and 3'. They are indicating where the bonding is occurring. This is 5', this is 3'. In between you have adenine bond with thymine, cytosine bond with guanine. But you need to remember that adenine always form bond with the thymine, cytosine will always with guanine. Similarly, you can write in the any sequence this one this way, right? And in this we know that adenine always form bond with thymine and guanine will form bond with the cytosine. So if you know the base strand of the one DNA, that is adenine, thymine, so on, cytosine, guanine, then T, then A, and they are asking about the another strand, then you can easily tell, yes, this is A, then this is T, this is T, this is A, G, C, A and T. So if you know one strand, you can easily tell about the another strand. So they are complementary to each other. Now, what are the differences between the deoxyribonucleic acid and ribonucleic acid? Now, the sugar present in the DNA is 2-deoxyribose, because on the second position there is no O, and sugar present in the ribonucleic acid is beta-D-ribose. Helical structure is double-stranded, single-stranded. DNA is a chemical base of heredity, and RNA is responsible for the formation of protein. Though it is responsible for the formation of protein, but the message is coming from the DNA only, because DNA to RNA and RNA to protein forms. In DNA you have ATGC, that is thymine, guanine, cytosine, adenine, while in case of RNA, instead of thymine there is uracil, that you need to learn. Then DNA occur only in the form one form. Here it will be, it is in the one form, and here it is in the three form. M stands for messenger RNA, T stands for transfer RNA, and R stands for ribosomal RNA. Ribosomal. So there are three types, there are one type. Now this is the structure. Now this is RNA and this is the DNA structure, double helical structure. RNA is a single stranded. This is RNA, which is single stranded, and DNA is a double stranded. And this we need to learn, remember always that in RNA is adenine, uracil, and guanine, cytosine, while in case of DNA, adenine always form bond with thymine, and guanine they form bond with the cytosine. There is no uracil in case of DNA. Now, what is the biological function of the nucleic acid? DNA is the chemical basis of heredity that we already discussed, and it is regarded as the reserve of genetic information. It is also responsible for maintaining the identity of different species of the organism. Like if we found the fossil of any organism or any species, then we can detect with the help of the DNA that what it was and what it is exactly belongs to. And DNA is capable of self-duplication and DNA transferred into the daughter cell. Because DNA is replicating and duplicating themselves, they are forming number of the daughter cells which are again forming the DNA. Nucleic acid is responsible for the formation of protein in the cell, but the message for the synthesis of protein is again present in the DNA. So mainly the DNA responsible for the heredity and number of the important things, like in the serials also, you can say in CID also, that they are detecting that the dead bodies also, and they are relating with the help of DNA that belongs to the which relations or the other things. Thanks for the day. This was the chapter of the biomolecule. I hope you understood.